My name is Andrea Johnson, and this is for Communications 100-6W. My topic is the conservation of the Atlantic horseshoe crab. When was the last time you received a vaccine? If it was within the last 40 years, you probably didn't know that you had a horseshoe crab to thank for that. Although conservation efforts do exist today, humanity needs to put more effort into finding a viable replacement for horseshoe crab blood and stop harvesting them before we push them to the point of extinction. A quote I really appreciate by Gerald M. Carborn, published in May of 2001 to the Providence Journal, states, "It's difficult for humans who have been around for a mere 3 million years to gain perspective on 250 million years. Horseshoe crabs predate dinosaurs by 60 million years, and still they endure, though their population is plummeting." I am not an expert in the field of marine biology. However, my references are. They've had articles published in the Journal of Clinical Pharmacy and Therapeutics, Science News, and the American Institute of Biological Sciences. First, we will take a look at what a horseshoe crab is, then why and how they are harvested, and the problems created by collecting them. Afterwards, I will go over current conservation efforts that exist to protect the horseshoe crab, finally concluding with viable solutions for eliminating horseshoe crab harvesting. To understand the horseshoe crab, you first have to understand that it's not a crab. It's not even a crustacean. Its scientific name is Xyphosera, which is a fancy way of saying they're more related to spiders and scorpions than they are crabs. The crab's tough exterior shell is covered with 3 separate pairs of eyes, 2 ventricle eyes underneath, and 2 on their tail. For this topic, I will be focusing solely on the Atlantic horseshoe crab, whose mating grounds happen to be the Delaware Bay. Harvesting horseshoe crabs from the Delaware Bay for use in the field of biomedicine is detrimental to their existence and continued survival. According to a U.S. News and World Report article published in July of 1999, written by Laura Tangley, a professor in the field of biomedicine, she states, in 1950, researchers discovered that horseshoe crabs' blue copper-rich blood contains a type of white blood cell that attaches to toxins produced by disease-causing bacteria. Known as LAL, it is now widely used by pharmaceutical industries to screen intravenous drugs for these bacteria. To produce LAL, a handful of companies collect and draw blood from large horseshoe crabs, returning them to the wild after 72 hours. The problem with this is the mortality rate is between 15 and 25% per extraction. Extraction is roughly 500 crabs. Harvesting entails turning them upside down and strapping them to a metal shelf where a needle is inserted near their heart and they are bled of approximately 30% of their blood. The researchers argue that this amount of blood loss is not fatal to the horseshoe crabs. Female crabs that are harvested are much less likely to mate when they're returned to the sea, or they die from stress or exhaustion, or the inability to fend for themselves in the wild. Sometime during the late 80s, we learned that humanity was threatening the horseshoe crab in a much more devastating way. Fishermen in the Delaware Bay had been collecting horseshoe crabs in astonishing quantities as a cheap alternative bait for predatory snails. During that time, a census was conducted and a 90% drop in the population was recorded. Although it was not the horseshoe crab that alerted scientists to the problem and prompted the investigation. Red knots are the migratory shorebird that alerted us. For the red knots, it is stated in a quote taken from an article, "The Effects of Horseshoe Crab Harvest in the Delaware Bay on Red Knots," published February 2009 to Bioscience by Lawrence J. Niles. For the red knots, it is the final stop before a single flight to Arctic breeding grounds, where on arrival in early June the weather is uncertain and feeding conditions are poor. Therefore, body reserves gained on the Delaware Bay are crucial for both the flight to the Arctic and survival and successful breeding. From the publication The Life and Time of a Living Fossil, published in February of 2005 by the American Institute of Biological Sciences, written by David R. Smith, he states, before 1999, conservation management were inconsistent and highly localized. Loss of spawning habitat has prompted fishery managers to formulate a coast-wide management plan, the Atlantic States Marine Fisheries Commission of 1998. Since the book's publication, harvest in the Delaware Bay region has been reduced to 25% of the peak harvest that occurred during the 1990s. Similarly, in 2000, the Carl N. Shuttner Jr. Horseshoe Crab Reserve was founded to aid in protecting the horseshoe crabs that spawn at Delaware Bay. Aside from fishing regulation and alternative forms of bait, it has been expected that the LAL test might someday be replaced by a genetically engineered replacement or another biochemical alternative. However, it is fair to question how much effort is being put into finding a viable solution when you consider laboratories make massive profits off harvesting. A pint of horseshoe crab blood is valued at roughly $15,000, and there are only 5 independent companies that are able to harvest horseshoe crabs and sell an extraction of their blood. To date, there is no replacement for the accuracy of the LAL test. However, scientists are looking for a solution. One example is the efforts in the MonoMac6 assay, as described in the Journal of Clinical Pharmacy and Therapeutics, published in August of 2000. The monoclitic cell line MonoMac6 is an alternative assay for detection of MonoMac6. The MonoMac6 was far below the threshold endotoxin limit described by the European Pharmacopoeia. So in other words, we have the technology and scientists are making the effort. Again, although conservation efforts exist today, humanity needs to put more effort and more funding behind finding a viable solution sooner. In conclusion, we took a look at what a horseshoe crab is, when and how they are harvested and why, and the problems created by collecting them. We went over what alerted scientists to the problem and what current conservation efforts exist today. Finally, concluding with insight into viable solutions for eliminating horseshoe crab harvesting. Horseshoe crabs are often referred to as living fossils. Not much has changed about them in over millions of years. After all that they have endured and outlasted, they face a threat that could push them to the point of extinction. At a 15 to 25 mortality rate is unacceptable for a species that has already had 90% of its population wiped out. What will be the cost if we push the horseshoe crab to the point of extinction before we have found a viable replacement to the LAL test? What could be the global ramifications of losing red knots and other migratory shorebirds? Better to wonder and not have to find out. The time is now. Speak up for those who have not evolved voices to speak for themselves. Let others know about the plight of the horseshoe crab, and together we can save these ancient wonders.